Hello friends, welcome to the IRCO channel. So today we are going to cover different factors regulating the development of herbicide resistance in weeds. So as you know that in today's scenario, majorly the weeds are developing resistance against the herbicides which was earlier susceptible to that very herbicides. And here we will see what are the different factors by which the weeds will develop the resistance against the herbicides, and that is why, as we know, that majority of the crop loss is due to the competition with respect to the weed. So here we will cover three factors. So majorly there are three factors which will be related to either it is weed factor, or the factor related to herbicide, or the factor related to crop cultivation practices. So we are going to cover these three factors one by one. So first factor, that is the weed factor. That is, in case of weed factor, so we are having first one is initial frequency. So initial frequency means you have to know that the number of resistant individuals which will exist in the natural population of a weed species. As we know that in a natural population, there are different weed species. There are different weed species: weed species 1, weed species 2, weed species 3, weed species 4, weed species 5. Accordingly, there are different types of weed species are there. And whenever you are applying any herbicide, to this field, might be some of the weed species will be got killed, whereas some of the weed species will may survived. So here in this case, what is the initial frequency of the natural, what is the initial frequency or the resistant individuals, what are the number of resistant individuals present in that natural population of a weed species. So here it is called as the initial frequency. So obviously, if higher the initial frequency will be there, then there will be greater or quicker chance of resistance development will be there in this case. So on an average, we have seen that 1 into 10 to the power minus 6. That means out of 1 million, one weed species may have a resistance against the herbicide. This is called the initial frequency. And this is the second factor we are going to cover, that is the hypersensitivity of the susceptible population of a weed to herbicide. Hypersensitivity means what? So that means 92 more than, for example, 92 95% susceptible population will be killed in single application of the herbicides. That means if there will be 10 number of population, if there will be, if there will be 20 number of populations is there, so in this case approximately 90% populations will be got killed. 90% population will be got killed, whereas the rest 10 to 5% population will get develop some resistance. That means whenever there will be hypersensitivity, means here more amount of susceptible population is there. So as more amount of susceptible population is there, so that means here the herbicide is highly effective. So here the herbicide is highly effective. So as you know that this is the natural selection. So by means of natural selection, if herbicide is very highly efficient to control the weeds, that means your weeds are highly susceptible to that herbicides, and these weeds will be killed very quickly. Hence, in this case, the selection pressure is very high. Hence, the quicker resistance development is there. So you have to remember, the theory is that if the selection pressure is very high, then the resistance development will be more quicker. So here quick resistance development will be there. So this is called the hypersensitivity of the susceptible population. So for example, suppose herbicide H1, it will kill 90 to 95% of the weed species, whereas suppose herbicide 2, it will kill only 60% of the weed species. That means here herbicide 1, because of the hypersensitivity of the weed species, so with respect to resistance development, the weed species, the weed species which is already susceptible to this herbicide 1, it will develop resistance more quickly as compared to the second herbicide. Okay, then next coming to third factor, that is the selection pressure. So here we can find out the selection pressure. What do you mean by selection pressure? So it is the relation between resistant and susceptible individuals. Suppose I am taking one plot. Suppose I am taking one plot, and in this plot there are some susceptible population, there are some susceptible population, and there might be some resistant populations. So suppose this is the resistant population, and these the red colors, these are the susceptible population. So selection pressure means the relative proportion of the live resistant population and susceptible individuals. So selection pressure, here I can write selection pressure means what? It means the number of population of live resistant population divided by susceptible individuals. This is the relative proportion or the ratio between the resistant population and the susceptible population. So as you know that if the selection pressure is very high, the theory says that if the selection pressure is very high, then the quick resistance development will be there. So you will see here, if it varies across herbicides. So this selection pressure depends upon the what type of herbicides you are using, and also across weed species. What are the different weed species? As you know that the different weed species will we can see their different weed species can act differently against the herbicides. Second thing is that application of high efficient herbicides. Earlier we have seen, if your herbicide is very highly efficient, that means there will be quick development of resistance. And if your herbicide is highly efficient and having single target site and very specific mode of action or same class of herbicides, then there will be high selection pressure. So we will see this part in at the herbicide factor also. We will cover it in detail. Next is the frequent application of without rotation. Suppose we are using same herbicide repeatedly. This H1 this year, next season this H1, next season the same herbicide is used. So due to continuous use of the herbicides, the resistance development will be very high because of the high selection pressure. Then herbicide with long residual activity. So in this case, herbicide, if the residual nature of the herbicide is very high, then also it exerts high selection pressure. So all this part we will cover when we will study, when we will read about the herbicide factor. Next one is pre-emergence herbicide exert high selection pressure as compared to the post-emergence herbicide. Hence, the quick resistance development is there against the pre-emergence herbicides. Then next, continuous monoculture. Monoculture means raising of one crop year after year. Suppose I am taking rice, then followed by rice, followed by rice. So as I am taking the same crop, this is called as monoculture. So in this case, you are using the same herbicide as you are having the same crop. So you are also using the same herbicide. Hence, there will be quick development of the resistance development. So similarly, zero tillage also exert high selection pressure. So all these things we will cover in the herbicide factor as well as in the crop cultivation practices factors. Then the next one is biological fitness of the weed. So as you know that, so in this case, suppose I am saying that one resistant weed is very much fit. Very much fit means what? Biological fit. Biological fit means what? It will produce more amount of seeds, more vegetative growth, and more amount of seeds. That means the population of this resistant weed will increase because of its biological fitness. That is why biological fitness, it is the relative evolutionary advantage of a phenotype based on its survival and reproductive success. Okay, so this means biological fitness will measure the potential evolutionary success of a phenotype, whether one weed will show resistant or it becomes susceptible. So it depends upon its biological fitness. So how fit? If the resistant weeds is there, that means more reproductive success is there. Again, though you are applying some herbicide, there will be no effect over the resistant weeds because of their biological fitness. So as well as depend growth rate, so which is related to the vegetative growth, photosynthesis, and this vegetative growth or growth rate is directly depend upon the high photosynthetic rate, and which result into the higher biological fitness to some individuals of the population of a weed. So in a population of weed, some weeds having resistant in nature because of their high reproductive growth, high vegetative growth, and they can survive and they can reproduce. Again, if you are applying the herbicide, still there will be no effect about these weeds because of their biological fitness. Then next, coming to weed biology. So whenever we are considering the weed biology, so in this case, first one will be seed dormancy. So in this case, seed dormancy, as you know that it is, we can say dissemination. Seed dormancy is a dissemination with respect to time. Dissemination with respect to time. Suppose, so today I am applying one herbicide, and this herbicide will kill those weeds which are germinated. It will kill those weeds which are germinated. That means there are some seeds which are not germinated, and there are some seeds which will germinate. So this herbicide will effectively kill these weed seeds which is germinated ones. But whatever the seeds which is not germinated, because of their seed dormancy nature, because of this, the feature is that is the seed dormancy. Now the seeds will not germinate as and when you are applying herbicides. At the time of herbicide application, whatever the seeds which are not germinated, these seeds in the successive season they develop resistance against that herbicides. Next thing is germination. So here it is directly related to the seed dormancy as well as the germination. Next one will be the mode of pollination. So high mode of, you know, there are different types of mode of pollination. Either it may be autocory or it may be allocory. So here the mode of pollination, there is the here you can go for either self pollination or you can go for the cross pollination, and this mode of pollination is directly related to the seed production capacity. So here the seed production capacity, if it is very high, then in this case the resistant population will, the quicker development of the resistant population will be there. Then again, seed bank which will act as the buffer. What is the seed bank? And as you know that in the inside the soil system there are different types of seeds of the weeds is present, which will produce further by means of germination, which will increase their population season after season. So this is called as the seed bank, and which will act as a buffer. Buffer means, so whenever is we have already seen, because of the seed dormancy, the seeds which will present in the seed bank in future they develop resistance. Then the next one, the last one, which is under weed factor, that is mode of inheritance of the resistance genes. As you know that mode of inheritance, there will be two types. Either it will be controlled by single gene or very few gene, or mode of inheritance by means of multiple gene. Always whatever the character which will be transmitted by the single gene, it is more effective in nature. Okay, that is why most of the herbicide resistance selected at the crop fields, whatever we have seen, they are mostly regulated by the single gene and having very high dominance in nature. These genes are dominant in nature, though we have seen the recessive gene also, but mostly the cases we have seen the highly dominant genes. Then coming to second one, so we have already covered the weed factor. Now coming to herbicide factor. So herbicide is the prime factor. You have to remember this one. Out of this three factor, that is weed factor, herbicide factor, and crop cultivation practices factor. So out of this, herbicide is the prime factor for the development of resistance in the weeds. So under this, we will cover, there is the application of herbicides which is highly effective in nature to the existing weed species. Obviously, we have seen that if H1 herbicide, another is H2 herbicide. So if H1 herbicide is highly effective, means it will kill 90 to 95% of the susceptible population of the weed. But H2, which are having low efficiency, maybe it will kill 60 to 65% of the susceptible population. So here, as this H1 is highly effective, so in this case, whatever the population is there, they are highly sensitive, or we can say hypersensitive in nature. These weeds are highly sensitive in nature. So though 90 to 95% species will be killed, but whatever the remaining 5 to 10% which will survive, so these weeds will develop resistance very quickly. These 5 to 10% of weeds which will survive, they will develop the resistance against the herbicide H1 very quickly, because in this case, if your herbicide is highly effective, so in this case it will exert high selection pressure, which will leads to quick resistance development. Okay, then next coming to second one, herbicides with a single target site and very specific mode of action, which are having of maybe same class of herbicide group, which will impose high selection pressure and leads to quicker resistance development. So let us come to an example. So we can give an example. So it says that herbicide with a single site target site or very specific mode of action. So suppose I am taking two types of herbicides. One will be, suppose will be isoproteron. Suppose I am writing the isoproteron. So isoproteron, it comes, this is a herbicide which come under a group that is phenylurea. So isoproteron is an herbicide, comes under group phenylurea, and it has having very specific mode of action that it will inhibit, it is majorly inhibit the photosynthesis process at PS2 site. So this is very specific mode of action. Very specific mode of action. But the second example I am taking, there is a pretilachlor. That is the pretilachlor. And pretilachlor, it is comes under the group that is chloroestamide. It comes under the group that is chloroestamide, and it will, the mode of action will be two. So in this pretilachlor, there will be two mode of action. Either it will inhibit the cell division, or it will inhibit the acetyl coenzyme carboxylase, that is ACCase enzyme, which leads to which will inhibit the fatty acid synthesis. That means here you can see there will be, I have taken two herbicides. One is isoproteron, another is, one will be isoproteron, another will be pretilachlor. So here it says that, the statement says that whatever the herbicides which are having single target site and very specific mode of action, it will exert high selection pressure and leads to quick resistance development. But whereas the herbicides with having different types of mode of action, it will provide low selection pressure, hence less rate of resistance development, or slow resistance development is there. Okay, I hope you are getting my point. Then the next factor that is, we can see there is a frequent application of herbicide without rotation or alteration or combining with other herbicides of different mode of action, so which will favor the selective evolution of the resistant biotypes of the weeds. So this is very simple. So suppose if I am applying isoproteron, in the season 1. In season 1, isoproteron is majorly used to control the Phalaris minor weed. In which field? In the wheat field. So suppose in the first season, I am using the isoproteron to control the Phalaris minor. In the second season also, I am using this isoproteron. In the third season, I am also using the isoproteron. In the fourth season, I am also using the isoproteron. So if I am using this same herbicides without any changing, without any alternation, or without any combination with the other herbicides of other mode of action, then what will happen? This Phalaris minor, so suppose in the first season, this Phalaris minor is now susceptible in the first season, but perhaps in the fifth season, this Phalaris minor has developed resistance against the isoproteron. Hence, in the first season, that Phalaris minor which was killed, now this Phalaris minor is not killed. So it is resistant against the isoproteron. So this is called as the frequent application of herbicides without rotation or alteration. Okay, or combination with the herbicides with a different mode of action. So that means, so suppose I am, so examples, I am taking another example. Suppose I am using the pretilachlor. Suppose I am using one that is pretilachlor. Pretilachlor. So suppose I am using pretilachlor. In other cases, I am using pyrazosulfuron ethyl. Pyrazosulfuron ethyl. So there will be two examples, pretilachlor and pyrazosulfuron ethyl. The pretilachlor will control the, already we have seen, it will control the weeds by either inhibiting the cell division process, or it will inhibit the fatty acid synthesis process by inhibiting the ACCase enzyme, acetyl coenzyme A carboxylase enzyme. Okay, so pyrazosulfuron ethyl, it comes under group sulfonyl urea, and it will inhibit the acetolactate synthase enzyme, that is acetolactate synthase enzyme, or acetohydroxy acid synthase enzyme, acetohydroxy acid synthase enzyme. So these enzymes are used to prepare branched chain amino acid, that is leucine, you can write leucine or isoleucine. Okay, so I have taken, suppose I am now taking the in the first season I am using the pyrazosulfuron ethyl. In the second season I am also using the pyrazosulfuron ethyl. Third season I am also using the pyrazosulfuron ethyl. So what will happen? If I am continuously using this pyrazosulfuron ethyl, then the weed which is earlier susceptible in nature, so now that weed becomes resistant against the pyrazosulfuron ethyl. But if I am in the first season I am applying only pyrazosulfuron ethyl, in the second season I am using a mixture of pyrazosulfuron ethyl plus pretilachlor. As we know that mode of action of pyrazosulfuron ethyl is to inhibit the branched chain amino acid by inhibiting the acetolactate acid synthase enzyme or AHS enzyme, whereas the mode of action of the pretilachlor is inhibiting the cell division and fatty acid synthesis by inhibiting the ACCase enzyme. So as you are applying or you are so here I am not altering. So here in the first season I am applying pyrazosulfuron ethyl, but in the second year onwards I am using a combination of pyrazosulfuron ethyl and pretilachlor, which are having different mode of action. So in this case there will be less rate of resistance development in the wheat. Okay, so that is why whenever you are using the herbicides, either you can change the herbicides. So today we are using the, suppose I am using here pyrazosulfuron ethyl. So in the next season I can using the bispirivec sodium. So I can change this herbicide from pyrazosulfuron ethyl to bispirivec sodium, or I can use this pyrazosulfuron ethyl but along with other herbicides of mode of action, that is pyrazosulfuron ethyl plus pretilachlor. So in this case I can decrease the rate of resistance development in the weeds. Okay, then the next one is application of herbicides with longer residual activity. So as you know that though it controls, it means what, though the herbicides having longer residual nature can control the susceptible population effectively, Suppose I am saying the atrazine. Suppose I am saying atrazine. Atrazine which is having long residual in nature. So as the long residual in nature and this atrazine residue is present within the soil, hence obviously it will control the susceptible population effectively for a longer period. For a longer period. But the question is whether this atrazine can control all the weeds? No. So basically, suppose this same 90 to 95% of weeds becomes killed, but whatever the remaining 5 to 10% population which are already survive, these weeds, these 5 to 10% of weeds in the following season, it will influence the resistant development, and this resistant population will flourish more. Okay, so because of the long residual nature, so there will be quick resistance development will be there whatever the population is susceptible, the remaining survive population. And as we know that some of the population will getting susceptible and some of the population is getting resistant. So in this case, in the those susceptible population will be killed, but whatever the resistant population will be there, they will flourish more very highly, and they will their population will increase day by day. The next one is kind of herbicide and time of application and dose of application. So coming to time of application. So time of application, it says that whether it is pre-emergence or whether it is post-emergence. So it says that the pre-emergence herbicides which will impose a strong selection pressure or high selection pressure, hence there will be quicker resistance development against the herbicide in the weed, because these pre-emergence herbicides, they will impose high selection pressure. Why? Because they are controlling the weeds at the time of germination as compared to the post-emergence ones. So basically, if there will be pre-emergence herbicide and post-emergence herbicides, for example, let us taking pretlachlor. Let us taking suppose I am taking a pyrazosulfuron ethyl. So suppose pyrazosulfuron ethyl is used as pre-emergence. Another example, suppose bispyrivec sodium, and it is used as post-emergence. So that is pyrazosulfuron ethyl is whenever you are applying as pre-emergence, it is controlling the weeds at the time of germination. Hence, it will provide high selection pressure. It leads to quick resistance development. But bispyrivec sodium or post-emergence, so here already it will affect the vegetative stage of the weed. So when weed is already present at the vegetative stage and bispyrivec sodium is applying, hence it is getting low selection pressure and slow resistance development will be there. Similarly, dose of application. So dose means if you are applying with higher dose, then obviously H1, if you are applying, the optimum is 1 kg AI per hectare. But if you are applying 4 kg AI per hectare, that means you are increasing the dose, means you are exert more selection pressure and you want to kill the weed population very effectively, hence quicker resistance development will be there. Okay, and there will be kind of herbicide means whether you are applying single only pretlachlor or sole pyrazosulfuron ethyl, or whether you are using both combination of pretlachlor and pyrazosulfuron ethyl. If it is single mode of action, then there will be quick resistance development. But if you are applying a ready mix herbicide or herbicide mixture with different mode of action, then the resistance development will be slow. The last factor which will affect the resistance development, there is the crop cultivation practices and cropping practices. So cultivation practices, so in this case first one is the tillage practice. So tillage practice, there will be two types of tillage, whether it is zero tillage or whether it is conventional tillage. So as you know that conventional tillage means what? Conventional tillage means you are manipulating the soil, and means in this case whatever the seeds which are present in the deeper layers, so you are taking to the upside, and these seeds will be exposed to the sunlight. So in case of conventional tillage, whatever the seeds which is present in the deeper layer of the soil, they will be now getting exposed to the sunlight and they will be killed. So conventional tillage is a practice to control the weeds itself. But what about the zero tillage? So in case of zero tillage, what you are using? The whatever the weed seeds present in the deeper layers. So this is the, so suppose what in this is the zero tillage. So in this case there are some weeds which is already present, over the soil, and there are some seeds or vegetative propagules which is present in the deeper layer, of the soil. And what is the procedure to control the weeds in the zero tillage? So to control the weeds, to control these weeds, what you are using? You are using the herbicide. Majorly you are using the glyphosate. That is why it is called as chemical plowing. That is why it is also called as chemical plowing, which is either you are using the glyphosate or you can using the glufosinate AM. So majorly in case of zero tillage, we are controlling the weeds by application of herbicides. So these herbicides will control majorly the surface weeds. These herbicides will control the majorly the surface weeds, or the weeds which is already present, and the surface weeds will be killed. But these deeper layer seeds, will not be killed. These will not killed. Hence, in successive generation or in the following season, these weed seeds will develop resistance against the herbicides, and they produce more and more seeds, and with vigorous reproductive growth will be there. So you can see here, zero tillage enhance the rate of resistance development because herbicides used here only kill the susceptible individuals whose seeds lie on the surface layer. What about the deeper layer? The deeper layer seeds which will survived in the previous season develop resistance and flourish into a greater population in the successive seasons. Hence, in case of zero tillage, the resistance development will be more quicker as compared to the conventional tillage. Then the last one, that is the monoculture. Monoculture means you are taking the same crop year after year or season after season. For example, suppose I am taking the wheat, then I am also taking the wheat, also I am taking the wheat in the successively. So in this case, suppose in wheat, what is the associated weed? So here the associated weed is Phalaris minor. And to control this Phalaris minor effectively, I am using the isoproteron. So that means as you are taking the same crop year after year, the crop bound weed or the crop associated weed will come along with the crop, and to control these weeds you are using the same herbicides year after year or season after season. That means as you are applying same herbicides, that means you are using high selection pressure. Hence the weeds, that is the Phalaris minor, you will be now resistance after some year. This Phalaris minor becomes resistance against the isoproteron. So this is the different factors we have covered with respect to the resistance development in weeds. So let us just summarize it. So here I have written that what are the factors which will affect the resistance development. So here we have covered three factors: weed factor, herbicide factor, and crop cultivation factor. So under the weed factor, first one is the initial frequency. If here we have seen high initial frequency means high selection pressure. So in this case, quick resistance development. Hypersensitivity means hypersensitivity means what? The weeds are more susceptible to a herbicide. That means here your herbicide is highly efficient, and high selection pressure leads to quick resistance development. Here next one is selection pressure. This is the relative proportion of resistant weed and susceptible weed. High selection pressure means quick resistance development. Then biological fitness. So this will decide the evolutionary behavior of a phenotype. Then weed biology, it leads to whether seed dormancy is present or not. The seed production is very high or not. Then coming to mode of inheritance, basically single gene is here in this case the herbicide resistance is controlled by majorly single gene, and it is a dominance factor will be there in the most of the cases. Okay, then coming to herbicide factor. So in this case we have covered under the herbicide factors. First one is highly effective herbicides. So if you are using highly effective herbicides, it will impose or exert a high selection pressure, leads to quick resistance development. Then next, mode of action. If you are using the single mode of action or very specific mode of action and single target site, then high selection pressure and lead to quick resistance development. Then next, frequent use of the same herbicide without rotation, without alteration, or without mixing with the other herbicides of different mode of action. So in this case, high resistance development will be there. Then long residual herbicides like atrazine. So if you are using long residual herbicides, then obviously whatever the resistant populations are there, they will flourish more. Then kind of application, time of application, and dose of application. So in this case, pre-emergence herbicides, as it will control the weeds effectively and at the germinating stage, hence it will impose high selection pressure, leads to quick resistance development. And high dose of herbicide means you are applying high selection pressure, hence in this case resistance development will be more quicker. Hence whenever you are using the herbicides, what should be the recommended or optimum dose you have to use it. Then the last one is crop cultivation factor. So in this case, zero tillage and conventional tillage. Zero tillage, under zero tillage, because of the deeper layer of seeds are there which is not killed, and these deeper layer seeds in the successive season, it will flourish more and develop resistance. Then monoculture means raising of the single crop year after year or season after season, leads to more crop associated weed, and because to control these associated weed you are using the same herbicide repeatedly, hence there will be more resistance development. So I hope you have understood these points. So in the next chapter we will study about the mechanisms of the herbicide resistance in weeds. Thank you.